our first lecture in chapter 6. I have broken it down into 2 videos. So this first one is going to be the bulk of the content from muscle anatomy and physiology, which is muscle tissue and contraction. The second video is going to be dealing with kind of the second half of the chapter, which is going to cover body movements, which kind of correspond with what you were learning with joints at the end of chapter 5 in the last chapter. So also just to make note here, this is the chapter that goes along with the muscle list that you're going to be working on in lab. So just like you did working on the bone list, make sure you are getting comfortably being able to name your muscles, and then those attachment points, which we call the origins and insertions, and then the movements. So flexion, extension, which we're going to be talking about in the second video of chapter 6. So we're going to focus more on the kind of the anatomy, physiology of muscle tissue in lecture. Lab is going to be focused primarily on your muscle list. So naming your muscles, knowing their attachment points, and knowing their functions. Are they contract? When they contract, are they causing flexion, extension, abduction, adduction of whatever body part? So make sure you're kind of setting aside enough time to focus on both the lecture stuff while you're working on your muscle list stuff. All right, so to begin, we're going to take a look, just a brief review of our muscle tissue types. So going back to our chapter 3 tissues information, if you remember, muscle tissue was one of our 4 major types of tissue. And then we had 3 different types of muscle tissue. So we had skeletal, which is attached to your bones primarily. We have cardiac, which makes up the walls of your heart. And we have smooth muscle, which makes up the walls of your hollow organs, like the stomach and the intestine, esophagus, bladder, uterus, those types of things. So we're going to focus on the rest of this chapter. The muscular system focuses on the skeletal muscle. When you come back after the summer break or fall break or whatever you want to call it, you will be looking more at the cardiovascular system in 122. And so you'll learn more about the cardiac muscle there. And then when you get into like the respiratory system and the digestive system and the reproductive systems, you'll see that smooth muscle kind of come back in those organs. So for now, we're going to focus just on skeletal muscle, but we do know cardiac and smooth muscle exist. All right, so functions of skeletal muscle, since we have that up here, it obviously produces movement. So muscle tissue has the ability to contract. So that is unique to muscle tissue. Epithelial, connective tissue, nervous tissue, they cannot get shorter. That's what contraction means. So muscle tissues contract. This allows them to provide movement. It also helps to maintain posture. So I can hold my head upright, I can put my shoulders back, I can sit in this chair because my skeletal muscles are contracting. I don't necessarily have to be actively moving, right? So if I'm sitting perfectly still, other than my face and my lips, I am still contracting my skeletal muscles. And by doing that, it's holding my skeleton in this particular position. Going along with that, it helps to stabilize joints. So imagine if you're doing bicep curls, like this, I am particularly moving this muscle at this joint, at my elbow joint, but I have other muscles that are stabilizing my shoulder. I have other muscles that are stabilizing my chest and my abs. So again, this kind of background level contraction helping us keep our posture and stabilizing joints as we do particular movements. And then the last function that's listed in your text is producing heat. So think about the last time you were cold, is you shiver, you get contraction of your skeletal muscles. It's like a built-in heater, because we'll be learning towards the end of this video that when muscles contract, they burn a lot of ATP. So you learned about ATP back in the chemical chapter and the production of ATP in cell physiology. It is our molecular energy currency of the cell. So whenever we need the cell to do things, it's going to break down that ATP. Muscle contraction uses a lot of ATP, and one of the byproducts of ATP breakdown, not only is it allowing us to move the muscle, but it generates heat as a byproduct, just like when you are driving your car. We explode gasoline to produce the energy to drive your motor, but a byproduct of that is heat. Right? That's why we got to put water and antifreeze and radiator, all that motor stuff. You can think of your skeletal muscles like a little motor. So they feed on the fuel of ATP, which comes from the food that you eat. It gives us mechanical work, which is contracting the muscle with a byproduct of heat. All right, so let's take a look at the anatomy of muscle tissue. skeletal muscle tissue. So I have two different pictures. They're basically showing the same thing. So sometimes I like to give, various perspectives and views so you get comfortable with relationships of things, not just memorizing one particular picture, but know how things are in relationship to other landmarks. So in muscle tissue, there are 3 layers of connective tissues. These are called the mysiums. So we have 3 mysiums, if you will. So our outermost mysium is what's called the epimysium. So I'll circle that on both pictures. Epi means outer or upon. It is a connective tissue sheath that surrounds the entire muscle, the whole muscle as a whole. Think biceps brachii. There's an epimysium surrounding that whole muscle. All of the muscles we have, again, you can reference your muscle list to get the names of the actual muscles, are made up of smaller units, kind of bundles of cells, and these are called fascicles. So a fascicle is a bundle of muscle cells. Each fascicle is wrapped by that next mysium, which is called perimysium. So I'm underlining that here in these 2 diagrams. So epimysium around the entire muscle, perimysium around the individual fascicles. And when we take a look at an individual muscle cell, right, there's a whole bundle of those. The endomysium is what lines the muscle fibers. So we see that here, endomysium would be surrounding the individual muscle cell. So I'm going to draw it right over here. If we're talking about endomysium, it would be on this picture right around in there. Okay, so epimysium, the outermost layer, perimysium, the in-between layer surrounding the fascicles, and endomysium, the very inner layer surrounding the individual cells. So there's a lot of kind of circle-y things going on. So we have the whole muscle made up of a whole bunch of fascicles. Each fascicle is made up of a whole bunch of cells, and each cell, as we're going to be diving in a little bit deeper, is going to be made up of their own little tube-like structures called myofibrils. Okay. And then what's kind of cool is all of those mysiums come together, the epimysium, the perimysium, the endomysium, and they form the tendon. Tendons are the connection from muscle to bone. Where you learn back in the skeletal system, ligaments are the connection between bone to bone. So ligament is bone to bone, tendon is muscle to muscle, and the tendon is made up of those connective tissue sheaths, kind of post-muscle fiber. So the epimysium, perimysium, endomysium all extend past the muscle to connect to the bone. So let's zoom in, right? So here would be our single muscle fiber. So muscle cells are called muscle cells, but you might also see them being called a muscle fiber. So like in the little description here, muscle fiber. So we've pulled out one of those muscle fibers from a fascicle, and we're going to take a look at it. And if I'm going to draw like where the endomysium would be, I'll kind of draw it in an orangey color. The endomysium would be right outside the cell. Okay? So it would be a sheath. Imagine a sheath kind of surrounding, think like loose connective tissue. So imagine this cell is surrounded by the endomysium. It's not shown in your picture, but I'm drawing it in because a lot of times students confuse the sarcolemma with the endomysium. Okay, so I'm going to erase most of those so it doesn't get in the way of the rest of the labeling. But I'll leave the upper endomysium picture there. And the reason I did that is because this first thing that we're going to identify is what we call the sarcolemma. Now, because this is a cell, it's got a phospholipid bilayer, a plasma membrane, just like any other cell, but we're calling it the sarcolemma. So this is basically the same thing as your plasma membrane. Okay, so sarcolemma holds everything in, and then just outside of that is where you're going to find that endomysium. Okay? So then if we take a look at what's inside of the cell, it's cram-packed full of these little tube-like things called myofibrils. They're the contractile organelle. They are the things that make the cells contract. We can also see, it's not labeled here, but I'm going to label one. They're kind of these little green dot things. These are going to be your mitochondria. Skeletal muscles have a lot of mitochondria, and if you can think back to what we learned about them in chapter 3, mitochondria make ATP. So this is the fuel, this is the gas station, if you will. This is where the ATP comes from. The food that you eat the sugars, the fats, the proteins, they diffuse into the cells, all of your cells, but since we're talking about muscles, we'll just focus on muscle. So the food stuffs diffuse into your cell, the mitochondria break the glucose down into carbon dioxide and water with ATP as the byproduct. So then all of this ATP can just diffuse back into the cell and go right into the needs for powering those contractions. All right, and then we have the nuclei. Skeletal muscle fibers are multinucleate, so they could have hundreds of nuclei per cell because they're really long. A muscle cell or a muscle fiber is as long as the muscle is long. So they're huge cells. So if I'm taking a look at my bicep right here, if the tendon starts here and the tendon starts here, that's a good 5 inches. That means my muscle fibers in my bicep, each one of the thousands of muscle fibers that make up that muscle are about 5 inches long. So they're long, that's why they're called fibers. And they contain a lot of nuclei. All right, so that's the kind of more large scale anatomy of the cell. We have the sarcolemma surrounding everything. We have mitochondria and nuclei kind of smushed in between all of these myofibrils. So now we're going to zoom in on one of these myofibrils. It's a long cylindrical structure, long tube, and it's made up of overlapping protein chains. And the major proteins we're going to be taking a look at are our thin and thick filaments. Sometimes you might be seeing the thin filament called actin and the thick filament be called myosin. Either of those terms are fine. So thin and thick filaments overlap each other, kind of like how my fingers are doing here. So when a muscle contracts, your thin and thick filaments go towards each other, and when they relax, they kind of go back to resting state. So it is this repeated bringing together and overlapping of these fibers that causes a muscle cell to contract. And when you have a whole bunch of muscle cells contracting, your muscles are contracting. But it's not just randomly organized. So these thin and thick filaments are organized very specifically because we don't want them all in different arrangements and bumping into each other. We need them nice and linear, and so they're going to have a nice overlap, and we don't get any loss of function when they're bumping into each other. So let's take a look at how these are organized, and they are organized into a structure of what's called a sarcomere. Okay? So a sarcomere, yes, sarcolemma. Oh, yes. Okay. I just wanted to make sure you guys are using that word because it was not listed on this picture. So a sarcomere, it's in the notes, is just one of these little repeating units that make us up the whole myofibril. And they go from Z disc to Z disc. Oh, my line was not quite correct here. So one sarcomere, boop, is from Z disc to Z disc. So that's the boundary. Okay, on the Z discs, I'll draw, you know what I'll do? I'll draw one over off to the side, so I'm not drawing over the picture. So I'll try to use the same colors. So I'm going to have a Z disc over here, and I'm going to have a Z disc over here. And this is also something that you can do as you're practicing when you're studying. Once you start getting more comfortable with your sarcomere structure, draw a sarcomere, just like I'm doing here. Coming off of the Z discs, these are going to be your thin filaments. So they're just kind of shown in blue. So I'll try to keep along with that same pattern. Okay. So those are my thin filaments. I got my Z disc zigzaggy boundary, which is kind of nice because the Z disc zigzags. I got my thin filaments coming off of those. And then right in the middle, we have this region that's called the M line. So I'm just kind of doing a dotted line, kind of what they show there. So the M line is kind of that right there. We'll talk about what the H zone is after we label all of these other things. So that's the M line. Then coming off of the M line, so again, we have to have everything organized. This is where you're going to get your thick filaments. And so I'm drawing those in red because that's how they are in the picture. So your thick filaments are supported by the M line in the middle M for middle, so that's always nice. Your thin filaments are supported by the Z disc. And so we have these nice organized fibers that are overlapping each other, but not touching each other, not yet anyways. So in a resting muscle, they're just gonna be hanging out like this, okay? So those are the pieces, Z line, M line, thin filaments, thick filaments. Then we can start labeling different regions of your sarcomere, kind of the zones. Oh, there's the M line. I just now saw it kind of off to the center. So where we have all of the thick filaments, from just the length of the thick filaments, this is called your A band. Okay? So the A band is as wide as your thick filaments are wide. So we see that right here. The I band is just going to have thin filaments. Now, it crosses a Z disc, and I only am drawing one sarcomere, so I'm kind of bracketing into one that I haven't drawn yet, but these are called the I bands. Okay, and so we can see that right here, where the Z disc is right in the middle. So think of A band as being the dark band, right? Dark. And the I band being the light band. Because when you look at these, if you remember, one of the characteristics of skeletal muscle was that it was striated. It was stripy in the microscope. And those stripes are coming from this very specific organization of your skeletal muscle tissue. So alternating A bands, I bands, A bands, I bands, because we have hundreds of these sarcomeres laid end to end within a myofibril, and they're all organized together. And so here's your dark bands and light bands. And now we see exactly where they're coming from. It's from this very specific organization of your thick and thin filaments within a sarcomere. All right, and then that H zone that we see that I kind of drew through, the H zone is within the M line. It's just the lighter side. It's on either side of the M zone. So where you're within the dark band, the A band, but there's no thin filament. So if you'll notice, I'll try to draw it on this picture. Oops, it's not letting me go. So I'm within the A band, but there's no thin filaments. So that's what we call the H zone. Okay. So again, as you're working through this, I know it can be overwhelming. It's kind of abstract ideas of all these A's and I's and Z's and M's and stuff, but really you need to practice drawing these out, labeling these out so you are more comfortable with the terminology, because when we get to the physiology, how these work, we have to be comfortable with these terms. So if you need to take a break, you might want to pause the video, do some practicing of all of these things, and then come back to take a look at the physiology once we start taking a look at how these muscles contract molecularly. Okay, so here's a closer look at the sarcomere and a little bit more realistic view of what these proteins look like. So again, I'm not going to draw out a sarcomere because we already did that, but what I want to do now is focus on what we call the zone of overlap. So the zone of overlap is where the thick and thin filaments overlap. That kind of makes sense, right? So what's eventually going to happen that we are going to describe is on the thick filaments. If you'll notice, they have these little like bead-like head thingies. These are called myosin heads because the thick filaments are called myosin proteins. And so basically what they'll do, get it correct, is they grab onto the thin filament, they pivot forward, and that's going to move the thin filament towards the M line. So if we have our thin filaments here and our thick filaments are pulling on them, they're going to move and shorten the sarcomere. So when we get to the physiology of this, and there's going to be a couple of videos that I have the YouTube links embedded in the PowerPoints slides. So you go and download those and you can find those links and copy and paste them into your browser so you can see those YouTube videos. So it is these kind of little myosin heads that are reaching up, pulling on the thin filament and powering them centrally towards the M line. And that's what actually happens when a muscle fiber contracts. Okay. So the characteristic of the membrane, if you will, so that sarcolemma has, in the notes, it has these two terms, irritability and contractility. Irritability just means it can get electrified. It can transmit an electrical impulse, kind of like a nerve or a neuron. Same type of idea. We'll be seeing that in chapter 7. So the irritability is they have the ability to transmit electrical signals, and then contractility just means they can get shorter. So if we're going to start take a look at the irritability part first, we have to see, well, where does that information, that signal come from? How do our muscle cells get the signal to contract? Where does that irritability come from? And it comes from neurons. Okay, so here's a picture of your spinal cord. Okay, what we call motor neurons, the nerve cells that control your muscle fibers are called motor neurons. Their cell body, so if you remember, our neuron kind of looked like this, long axon. We had the cell body with a nucleus. So this part here is in the spinal cord. Okay, so that's these little circles, the purple and the pink circles over here. And then the long axons, those travel out from the spinal cord via your nerves, right? So if you heard of your brachial nerve, your ulnar nerve, your sciatic nerve, your femoral nerve, all those nerves carry these neurons, these axons, and then they go and communicate and get into very close contact with that sarcolemma. And that's where the signal is going to be received. And we're going to take a closer look at that. But first, we need to see, how is this control organized? How many neurons, how many muscle fibers can be controlled by a single neuron? Well, it depends. It depends on what body action you are trying to do. So if you're doing a big muscle contraction like this, you might have one motor neuron coming out. So we'll say it's the red one. We'll say this is motor unit number 1, the red neuron. So that signal is going to come out and it can communicate to, let's see, we have 3, 2, this one, and this one. So those 2 muscle fibers are controlled by the red neuron. Okay? And if I'm just going to need to contract my arm like this, I might just have to fire that one red motor neuron. And this is what's called a motor unit. A motor unit is the motor neuron and all the muscle fibers that it communicates to. So in this case, the red motor unit comes out through your nerve, goes to your muscle, splits. In this case, it's going to communicate to 2 muscle fibers. So if I'm not lifting anything, I'm just contracting my arm, that's all the weight that I need. That's all the contraction I need to lift just the weight of my arm. Let's see, I got to find something heavier. So let's say I'm going to now add some weight. So this is just my binder for 121. So now if I want to contract my muscle, lifting the weight of my binder, I might have to fire another motor neuron. So I'll use the purple since it's purple. So motor unit 2 fires this, connects to this, this, and this. So that motor unit connects to 3 muscle fibers. So if I need to lift a lot of heavier weight, I might fire both the red and the purple motor units, contracting all 5 muscles, gives me more tension. I can overcome the resistance of my binder and lift my binder up. Okay? So that's what a motor unit is, all of the muscle fibers that a single motor neuron controls. Okay? So we're going to take a closer look at that, what's called the neuromuscular junction. It is where these neurons, you can see them all right here, landing on their individual muscle fibers. Okay, so here we have some more pictures of this neuromuscular junction. So we've zoomed way into just right here on the motor neuron and the sarcolemma. We kind of have these little indentations. We can see them, the I just lost it. The electron microscope picture, that gray and white picture. So now what we're going to do is we're going to take a look down here and kind of identify some of the structures that we're seeing. So the yellow is the membrane of the neuron. Okay. Within that membrane, so let me draw, I'll just try to draw things over here so we can take it step by step. Okay. Within that very terminal end of the motor neuron, we have these little packages, they're called vesicles, filled with neurotransmitter. Looks like they're doing green circles, right? So pretend these are circles. I can't draw them super great on my PowerPoint here. So those are my neurotransmitters. These are chemical messages. In this case, it's called acetylcholine. So ACH stands for acetylcholine. Okay, acetylcholine is a neurotransmitter. And then on the, let's see, they're kind of doing a gray. I don't have gray. Let's do light blue. So here's going to be your sarcolemma. Okay, so that's the plasma membrane of the muscle. And on that, there are little receptors. I'm going to draw them dark blue, kind of like in that little picture here. Zoop. These are the receptors. So then when the signal comes, so let's say your brain says, contract your muscle. There is an action potential. I'm just going to draw like a little lightning bolt that comes down. That action potential comes down the surface of the membrane on the neuron, and it triggers those vesicles to exocytose. We learned that word back in chapter 3, right? So it's fusing with that outer membrane. And then what you end up with is all of these acetylcholine molecules in this space called the synaptic cleft. Okay, so here's our synaptic cleft. It's a space these membranes do not touch. So your motor neurons do not touch your sarcolemma. When those acetylcholine molecules bind to the receptors, so a couple of them are going to bind here. What they do is they open channels, right? Remember, we have membrane proteins. They can be channel proteins. So without the acetylcholine, they're closed. In the presence of acetylcholine, they open, and there are sodium ions. Looks like they're yellow. So we have all of the sodium rushing into the cell. And I'm going to do the darker color because that's really hard to see. There we go. So the sodium rushes in, and it's positively charged. So basically what we're doing is we are generating an electrical event along the sarcolemma. So the motor neuron signal comes down, it triggers a release of these neurotransmitters. They cross that little space, the synaptic cleft, bind to the receptors, open these channels, sodium rushes in, and we've just activated the sarcolemma. That's that irritability that I mentioned earlier. So now what? What do we do with this excited membrane? Well, it is called an action potential. Right? This box I just circled in yellow over here. That action potential runs down these tubes called T-tubules, and that runs right next to, do we have sarcoplasmic reticulum? We did not see it on that last slide. So sarcoplasmic reticulum is a modified smooth ER. So if you remember that, storage places, it stores calcium. And that calcium is going to be released when that signal travels down the T-tubule, and it's going to flood all over those thick and thin filaments. Okay, so now let's take a look inside of the cell and see what happens with that when that calcium shows up. Okay. So here I have a picture up on the top of a muscle fiber. So here's again, I'll just highlight the things that we should be familiar with. So here's our sarcolemma, or sorry, where's my sarcolemma? Here's my sarcolemma. That's the plasma membrane. Here's the T-tubule, these transverse tubules. So they're continuous. So that electrical signal that's traveling along the sarcolemma will travel all the way through these T-tubules that are completely surrounding every single one of those myofibrils, and they're jam-packed full of calcium. Okay, and then here's your myofibrils made up of all of these repeating units of sarcomeres. Okay, so in this picture, let's just take our zoomers and just zoom way in to just one interaction between a thick filament and a thin filament. Okay, so those little myosin heads that we saw, the thin filament has, let me see, make sure. There's some words that are used. I just want to make sure you guys are, I'm not going too deep into it. Okay, so on the thin filament, which is this one right here, kind of the purple beads, there is a binding site for the calcium, and when there's no calcium, the myosin head cannot bind, it's blocked. When the calcium shows up, the active site, the binding site is revealed, and so what happens is we make this cross bridge. So in the presence of calcium, the myosin head can reach up and grab onto that thin filament, and it's powered by ATP. And then when it kind of cocks its head forward, it kind of pushes this forward, it releases those breakdown products of ATP, which is ADP and phosphate. and then another ATP molecule comes in, breaks down, cocks the head back up. And as long as there's calcium and as long as there's ATP, your muscles will continue to pull those thin filaments toward the M line, and that's your muscle contraction. So it's kind of cool to know that you are learning the molecular way of how your muscles work. It's really quite fascinating. It's all of this chemistry. That's why you guys had to learn a little bit of chemistry at the beginning to make sure you are comfortable with ATP and sodium and calcium ions and these things that are needed to do these really fascinating physiological responses inside of your body. So this is called the sliding filament theory because before they actually knew all the molecular workings of this, under the microscope, they took tissues and they kind of electrified it, and they actually saw those thick and thin, they saw the A bands and the I bands kind of change shape in a muscle cell. And they're like, I hypothesized there might be some filaments in there sliding across one another. And then that has been supported by evidence. We now sometimes call it the excitation contraction coupling. Excitation is the arrival of the signal from the neuron. The contraction is this process that you see right here with the thin filaments pulling on the thick. And then is the T tubules and the calcium that couple those things together. Okay. So here's just a little cartoon to kind of show that idea that when your calcium, you have these little ATP powered myosin heads, they're ready to go, but they can't, it says they're ready to rock, but they can't get a good grip because the calcium hasn't showed up yet. So here comes the calcium, it moves the blocking parts away, they can grab hold, they do their power stroke, and they move the thin filament forward. So here's a couple YouTube videos. You can link, copy and paste those links from the PowerPoint file because it's always great to watch something like this in animation. So make sure you find those, or if you just want to search your own YouTube video of sliding filament theory, excitation contraction coupling, skeletal muscle contraction, all of those will get you some good results on watching some animations for this process. Okay, so now that we understand the molecular piece of how muscles contract, and again, if you're still struggling with that, you need to send me emails, ask Wade some questions, come to an office hour to get clarification on that, because that physiology can be a little bit more challenging because it's very cellular and molecular. So please reach out and get help if you are struggling with that idea. All right, so what we just talked about is happening inside individual cells, but we don't really work with individual cells. We work with our whole cell, our whole organs, right? My whole bicep muscle is contracting. So how do we control that? Well, there's a couple different ways that we do that. One, we've already talked about with the amount of cells that are being stimulated by your motor units. Am I just picking up a little piece of paper? Maybe one motor unit. If I'm picking up, a big heavy thing, I might be activating multiple motor units. The other way that we can increase the amount of tension that we're producing is send those signals faster. So if we send just one motor neuron firing, like pew, we just get one, our muscles contract and then they relax and we get a twitch. Like think of an eye twitch or a muscle leg twitch or something like that. You can't really do much with that. It's just like one little tink and then it's done. If your signals come in a little bit faster, take a look and you can see the tension is going up. And in that case, we can actually sum these. It's called summing of contractions, and we can actually get some work done with that. If they come in a lot faster here, we can see we are also increasing our tension, and we can produce even more tension, especially if we're doing this with more motor units. This is called unfused or incomplete tetanus, which is different than the disease. It's just more of a clinical term here. But if they're coming so fast that there's no rest, right? So take a look at our graph here. There's no reset period like we see in all of the other waves. This is just like constant contraction. And we don't really use this very often either, because if your muscle's contracted and it's staying contracted and there's no relaxation, you're not getting any work done because your bones are all tight. So most of our normal work is done between these two speeds, if you will, or frequencies of how fast these electrical impulses show up. So twitches don't let us do anything because it's just like one quick twitch. Diffuse tetanus doesn't do us anything because our muscles are tight and contracted completely. It is between summation of the waves and incomplete tetanus where we can generate a variety of amounts of tension based on how many muscle cells are being stimulated at those particular frequencies. All right. The last couple topics we're going to talk about. All right, so this next slide is covering where energy for muscle contraction comes from. Now, we talked earlier that ATP is the source of energy, and that's true, and that's not going to change because that's what drives the myosin heads pulling on the thin filaments. But what we are going to talk about here is where does that ATP come from? So there's three different sources, I guess you can think about it. Two of them are anaerobic, which means you do not require oxygen. But the downside to that is they don't last for very long. So the first one here in picture in kind of column A is something called creatine phosphate, which is a creatine molecule hooked to a phosphate group, which pretty straightforward there. And so if your cells need some really quick energy, some quick ATP, they can take that, the phosphate group off of here, add that to the ADP, and then you get ATP. And that can go back and forth, it's reversible. So as you have a lot of phosphate group, you can temporarily store that onto creatine to make the creatine phosphate, and then you can reverse that and donate that phosphate group onto the ADP, giving you ATP. But if you take a look, it only lasts for about 15 seconds. So there's not a huge supply. I'm going to go over to the third box here because this is another anaerobic, so oxygen use is none. This is just straight glycolysis. And if you remember, glycolysis produces 2 ATPs per glucose. So one glycolysis process only gets you 2 ATPs, and it lasts a little bit longer. So we're about 30 to 60 seconds. Because we have a lot more glucose, our muscles store glucose as glycogen in pretty large amounts. But still, we're running on like less than a minute's worth of this energy in an anaerobic condition. So this last column, this is where most of our ATP comes from via aerobic respiration. That was the whole glycolysis, Krebs electron transport chain, the cellular respiration process, that in the presence of oxygen, it's required, you can have hours of energy. And this is how we do our day-to-day activities. We really only get into just glycolysis or creatine phosphate when we're in this peak high intensity training where your breathing, your respiration rate is so fast that we cannot provide enough oxygen to drive the aerobic respiration. So this slide is just a little cartoon kind of showing muscle fatigue, and there's a video down here. Again, you can follow that link in Blackboard on the PowerPoint file to see a real life example of muscle fatigue. But you can get to the point where your brain's like, I want to move my muscle, but you've depleted all of your resources, or your oxygen level is not high enough to drive aerobic respiration. You've used up all your ATP from glycolysis and creatine phosphate. So the sarcomere is like, hey, we need more ATP. We can't do muscle contraction. And the mitochondria is like, I don't have enough of those raw materials or oxygen to make enough ATP. So then your muscles just don't contract. And that's true muscle fatigue. All right, the last couple slides here talk about just some vocabulary that you can be familiar with. When we talk about muscle contractions, there's 2 types. So the first one is called isotonic. So this is isotonic, is once you find a weight that you can lift, when you are contracting, your muscles get shorter and you are producing about a consistent amount of tension. That's the iso meaning the same, tonic meaning tension. As you contract, your muscles get shorter, and then when you let go, the muscles can get longer. So this is isotonic contraction, where this one over here is isometric. Again, iso meaning the same, metric meaning length. So in this case, he's pulling up on something that's bolted to the ground. So this would imagine you trying to push a wall or lift something that's 500 pounds. You can't, I guess, unless you're an Olympic weightlifter. But for somebody like me, there's no way I'm going to lift 500 pounds. But if I try, I'm still producing tension in my muscle. I can feel it. It's tense, but it's not changing length. So isometric is same length. Isotonic is same tension, and the length changes. Isometric is you're producing tension, but the length does not change. Okay. The last slide here, I tried to go a humorous route here with discussion of muscle tone. So this is a young Arnold and an older Arnold. Age definitely has a role to play in this. But when we talk about muscle tone, it's the background level of activity that your muscles just do without you actively contracting. So when you have high muscle tone, your muscles are partially contracted at this kind of background level. This is usually, we say somebody is toned or ripped or buff. Just when they're standing there, they look like they are contracting when they're not consciously contracting at the background level. If you are more soft and squishy and flabby, or some other terms that kind of go along with low muscle tone, is the background level of muscle contraction is a lot less. And so you don't have that firm look on your muscles when you don't actively contract them. All right, so that wraps up the first half. I know this is a lot longer of a video, so you might have had to pause and stop, but I just wanted to put all of the physiology together into one spot. So again, reach out if you have questions, rewatch the videos, go and follow those YouTube links I have posted in the PowerPoint files themselves, and I will see you next time for movements, body movements and muscle names. Okay, bye.